Ni hao, one page tutor here. So in this video I'll talk about plant cell versus animal cell and how I use diagram to know those organelle structure and function, because by knowing the organelle structure and function you'll know the difference between animal cell and plant cell. At the same time, by having that diagram you get to know the process of extracellular and intracellular production, because knowing the sequence of this production can actually make you understand more on the function of organelle. I'll teach you step by step on drawing animal cell and plant cell from this to this. Okay, so first thing, the outermost layer of animal cell and plant cell, it's obviously different because plant cell is it having a fixed shape, which is like a rectangular shape, while for animal cell it's not a perfect circle because it doesn't have cell wall, which is a strong structure to maintain a fixed shape of plasma membrane. So first thing, you have to draw from center the biggest organelle inside animal cell, which is the nucleus. So I will start off by drawing the nucleolus first, then chromatin. So as you can see that chromatin and chromosome are different. This is known as chromatin, this is known as chromosome. Chromatin is condensed, only can form chromosome because chromatin is not yet ready for cell division. It needs to condense to form chromosome only for cell division. So chromosome, you will only see chromosome in cell division chapter. So that's the difference between chromatin and chromosome. One is not condensed enough and another one is condensed enough until you could see under microscope. So you will have nuclear membrane. So this whole thing is called a nucleus. Another nuclear membrane would actually form by RER, because RER is actually surrounding the nucleus. So as you can see, there are holes between. This is the nuclear pore. So this is RER, since it's called as rough endoplasmic reticulum. Rough endoplasmic means that when you see like a surface has dots on top, it's not smooth, that is the ribosome. In exam, you have to write in full form. You can't write RER to get marks if the question asks you to name it. So beside RER would be smooth endoplasmic reticulum. These two types of endoplasmic reticulum, you can see that the similarities of these two is that they are connecting. Each sac is connecting to each other, not separating like what Golgi apparatus does. So this is the main thing to differentiate endoplasmic reticulum, the ERs, with Golgi apparatus. So next is transport vesicle. This is the Golgi apparatus. So when you're drawing Golgi apparatus, you can actually imagine you are drawing red blood cell. Red blood cell is it's a flattened sac by concave shape, and the middle part is like pinching inside. So while you're drawing this, it has like how a red blood cell looks. It would definitely ends with this shape. And after Golgi apparatus, it will be secretory vesicle and lysosome. So as you can see that the transport and secretory vesicle have the same size, while lysosome will have bigger than that two vesicles. Okay, after drawing this, you will have mitochondria at the side. While you are drawing mitochondria, please don't draw it bigger than nucleus. Nucleus must always be the biggest organelle inside animal cell. And also draw centriole. Centriole is something which is not found in plant cell. Centriole is the one you learn in cell division, the mitosis meiosis chapter. While we are drawing the mitochondria, remember there are two layers. One is inner, one is outer, and the inner layer would be highly folded to form cristae. Okay, now we have done drawing all the organelles inside. Let's label each of them and the functions. I'll talk about those organelles which is responsible on intracellular and extracellular enzyme. Okay, some of you might think that what is intracellular and extracellular? Enzyme is a protein when it is not yet modified. So when protein go through Golgi apparatus, it will modify into specific function, which is enzyme. So enzyme is under protein. And what is intracellular and extracellular? Intra stands for inside. So this is for the enzyme which is used inside the cell. While extracellular, extra means outside. So extracellular enzyme, when you produce, you have to release out of the cell for other cell to be used. So I'll talk about the organelles which involve in this process first. So while you're drawing, remember, don't let those arrows, don't let those labeling line to intercept each other, and don't draw arrow also. Don't draw arrow and don't intercept. Okay, so first thing, so these are the organelles which involve in production of enzyme. Enzyme is a protein. Inside nucleus there's chromatin and nucleolus. Chromatin is the genetic material which contains the genetic information. This is genetic material which contains the genetic information. Genetic info is like a message I ask ribosome to produce protein. That's why nucleus has the function of control cell activity, because it send out message. It's like a leader which send out message for other organelles surrounding nucleus to do the job for ribosome to synthesize protein. So at the same time, you know that nucleus has the function of control all cell activity. Okay, so genetic info is like a message. Genetic info for ribosome on RER to synthesize protein. So once the message is out, so it will reach to the ribosome on RER first. So the ribosome on RER will receive that message, which is the genetic info, and synthesize protein. So that is the site of synthesizing protein. Synthesize is like produce. Please don't mess up spelling. This is synthesize. Synthesis is this one. Synthesis is like noun. Site for protein synthesis. Synthesize protein. Synthesize is the verb. So many people actually thought that RER is the one who produced the protein. But actually RER is not that organelle which produce protein. Ribosome on RER is the one which produce protein. So RER is to package the protein synthesized by ribosome to transport vesicle. Okay, so remember that RER is just like a shelter for the ribosome. Ribosome is the one who wasi. Ribosome is the one who produce protein, because the protein produced, we won't know that maybe the environment inside cytoplasm might destroy the protein. So we need a vesicle to protect the protein form. So RER will package the protein, which is produced by ribosome, inside transport vesicle. Then it will transport to Golgi apparatus. So it goes like this. The chromatin send out the genetic info to the ribosome on RER. The ribosome will produce protein, then that RER will package into transport vesicle, then send to Golgi apparatus. Okay, now Golgi apparatus. Golgi apparatus will modify the protein into specific function, for example, enzyme. Because protein doesn't give any function to other cell. Enzyme will give function like in digestion or any other things. So it needs to modify that protein into specific function first. So it could modify into enzyme or into glycoprotein. So once it modify already, it will package into secretory vesicle. This one I've said that this is transport vesicle. So this is secretory vesicle. Okay, why do I use modify word and package word? Modify, this term modify means that it will enhance the function of protein into something useful, while package is like you put that substance into a protected capsule. So this secretory vesicle will bring the enzyme near to plasma membrane, so it can fuse with plasma membrane, then exocytosis to release enzyme out of cell. So this is actually for extracellular enzyme, because just now I've mentioned that extracellular is for enzyme to be used out of cell. So this enzyme is produced and released out of cell. So this is extracellular enzyme. So if you want to differentiate the vesicle in a better way, you draw the dots inside. Dots is like those substance are the protein or the enzyme. So it is released. Exocytosis is a process which is to release something. Okay, if you couldn't differentiate transport vesicle and secretory vesicle, you look at the word. Transport means is to carry something, right? So transport vesicle is to carry the protein form on ribosome to Golgi apparatus for modification, while for secretory vesicle is to bring enzyme form on Golgi apparatus to secrete out of cell. Secrete is like release, so it's released out of cell. So now we have done explaining those which are responsible for enzyme production. Now talk about those which are for other functions. Okay, let's look at this smooth endoplasmic reticulum. It doesn't have ribosome on top, so it's not for protein production anymore. It's for lipid production, lipid synthesis. So the main difference is one is protein, one is lipid. Okay, so next, as you can see from this picture here, which I draw it bigger, mitochondria has actually two membrane. Remember that this thing is called inner membrane. This is a common mistake for most people when the question asks like this: name M. And most people say that M is cristae, which is wrong. M is inner membrane, while inner membrane highly folded to form cristae. This in red pen is the function of inner membrane, is the function of mitochondria. So the inner membrane is highly folded to form cristae, while cristae having a large surface area, because it's highly folded to form a large surface area of cristae for and more energy production. Okay, next would be the centriole. Centriole is only found in animal cell. Okay, so centriole is something you learn in cell division, right? Centriole, which is at the site of animal cell, and it forms spindle fiber which hold the centromer of chromosome. It can arrange on the metaphase plate. So that is the function of centriole. Centriole forms spindle fiber in cell division. Okay, next is the lysosome. Lysosome also something which you only found in animal cell. Lysosome contains hydrolytic enzyme, or you can also know it as lysozyme. It's able to hydrolyze food and able to digest or break down the worn out organelles. So hydrolyze, digest, and break down are three terms which you can use it in replace each other, because three of them have the same meanings. So lysozyme, you also meet this word in amoeba, in the unicellular organism, when amoeba extend its pseudopodia and engulf food particles and form a food vacuole, and it release the hydrolytic enzyme, or in other words is lysozyme, to digest the food particles into simpler molecules. Something you could actually relate it to this, so you can remember what's the function of lysosome. Highlight those which you only found in animal cell: lysosome and centriole, and highlight those which is responsible for enzyme production: chromatin, or also nucleus, because nucleus contains those genetic material to produce the genetic information for ribosome to synthesize protein, the ribosome or RER, the RER itself, then to transport vesicle, Golgi apparatus, secretory vesicle, cytoplasm. And inside the animal cell, there are free moving ribosome. Ribosome not only found on RER, it also found inside cytoplasm, which is not attached to anything. So cytoplasm have the function of being a medium for biochemical reaction. So plasma membrane have the function of regulate movement of substance in and out of cell. So plasma membrane, you can actually relate to chapter 3. What's the function of plasma membrane there? Plasma membrane and cytoplasm, they are not organelle. Remember, these two are not organelle. If for plant cell, cell wall also not organelle. Okay, next is plant cell. So plant cell, you start off by drawing the outermost cell wall and the inner membrane, which is the plasma membrane. Then continue with the biggest organelle, which is the vacuole. So when you draw vacuole, it actually looks like this. It's like purposely have a bit of space for the nucleus to fit. So the nucleus, same thing like here. So draw the chromatin, nucleolus, the RER, and the rest of the nuclear envelope. Okay, this is nuclear envelope, and this is the hole is the nuclear pore. Near to those RER, there's SER. So Golgi apparatus must be next to RER. So flattened sac, and the final thing that would definitely be there, and only secretory vesicle, and here transport vesicle. Okay, continue by drawing the mitochondria and chloroplasts. So first thing, you have to draw the double membrane, 3 lines, and shade it in this arrangement. The shading is granum, while multiple stacks, one stack is called granum, multiple stacks are called grana, and continue by labeling the others which are also found inside animal cell, and secretory vesicle is between plasma membrane and Golgi apparatus, while transport vesicle, which I left out, is between Golgi apparatus and RER. And continue by labeling cytoplasm, cell wall, and the free moving ribosome. So let me draw a close-up of chloroplasts. So it looks like this. A few pebbles stack up together to form granum. If we're to, okay, just an extra info, one pebble, so-called pebble, is called thylakoid, but you don't have to know in SPM. You just have to know that granum is one stack of thylakoid is called granum, and the fluid inside chloroplast is called stroma. Stroma is responsible for dark reaction of photosynthesis, while granum is responsible for light reaction. So granum is the one that contains chlorophyll to trap sunlight, absorb sunlight, and convert it to glucose through photosynthesis. So if we're to ask energy conversion, sunlight is light energy while glucose is chemical energy. So convert light energy to chemical energy. So if we're to ask the adaptation of chloroplasts, you have to say that the granum is highly folded because there are a lot of thylakoids to form one stack of granum. It's highly folded to have higher surface area to absorb more sunlight. Okay, next, function of cell wall. Cell wall is made up of cellulose. Cellulose is a strong fiber. This strong fiber is able to provide support, which is a fixed shape, maintain the fixed shape of plant cell, and also provide protection to protect those organelles inside plant cell. Next is the function of vacuole. Vacuole is able to store water and nutrients. So the water, you can actually relate it to the osmosis chapter. So it stores water to have turgor pressure to provide turgidity to plant cell. As usual, highlight those which are only found in plant cell. I highlight in green color. We have finished drawing both of the cells. Now we can make a comparison like the difference and the similarities. First thing, what organelles are only found inside that specific cell? So like lysosome and centrioles are only found inside animal cell. If we were to talk more why lysosome only found inside animal cell, lysosome function is to break down food particles, is to break down something. Animal cell, like us human, we feed on others. So we need an enzyme to break down food particles. We need lysozyme. While plant cell, they can make food on its own, so they need chloroplasts. Chloroplasts can trap sunlight and convert it to glucose, which is their food. So chloroplast is only found inside plant cell. Cell wall is only found inside plant cells, so it can provide a fixed structure, while animal cell doesn't have a fixed structure. While for vacuole is to store water to have the turgidity in plant cell. So you can talk more. Don't only say like, oh, plant cell has chloroplast, cell wall, vacuole, while animal cell doesn't. You talk more about it. Talk about the function so you can secure the mark. Second is produce food on its own. Animal cell doesn't produce food on its own, while plant cell does. So like previously, you don't take lysosome and chloroplast. So in this second point, you can talk about the function of lysosome and chloroplast. Like animal cell doesn't produce food on its own because it doesn't contain chloroplast, which can convert sunlight into glucose. It contains lysosome, which can break down food particles using lysozyme. Third thing, shape of cell. Animal cell has irregular shape because it doesn't contain cell wall, but plant cell has a fixed shape because it contains cell wall. Okay, now the similarities between both cells. What organelle are found inside both? You look back, those which are not highlighted are those which are found in both. So like plasma membrane, nucleus, mitochondria. So you can talk more like why do both cells need plasma membrane? Because plasma membrane ensure particles to move in and out of cell. Nucleus is to provide the genetic material for protein to produce and modify into specific function, for example enzyme. Then third is mitochondria, which is to provide ATP for animal cell to have energy for movement, while plant cell to have energy for translocation or those. If you have any doubts or you need more example questions on this topic, you can leave in the comment section. Thank you so much for watching.